Welcome to the Deep Dive. We're here to cut through the noise, get straight to what matters, help you stay informed. And today, wow, we're diving into something pretty exciting. It's an area of science that's not just, inching forward, but really seems to be revolutionizing how we approach medicine. We're talking about drug discovery, specifically how AI, artificial intelligence, is just transforming the design of these really important therapeutics called antibodies. Our main source for this deep dive is a really fresh piece of research, super cutting edge. It's called zero-shot antibody design in a 24-well plate. Just came out, dated June 30th, 2025, from the Chai discovery team. And believe me, the findings are pretty significant. So our mission today is basically to unpack how this new AI model, they call it Chai-2, is letting scientists design functional antibodies, like totally from scratch. And they're doing it with incredible speed and efficiency. We're talking about making drug discovery faster, way more precise, and honestly, a lot more accessible than it's ever been. Okay, so let's unpack this.
Yeah, and to really get why Chai-2 is such a big deal, we kind of need to understand the old way, right? The world, it's changing. Antibodies, they're amazing biological tools. They've been around for, what, over 500 million years? Nature figured them out a long time ago. And what makes them so great for medicine is they're incredibly specific. They can recognize and, latch onto molecular targets with really high precision. Think of them as these perfect little molecular keys. So yeah, it's no wonder their use in medicine has just exploded. I saw a stat, by 2022, these things called monoclonal antibodies, they made up over half of all biopharma approvals in the US and Europe. That's up from just 20% back in the early 2000s. Huge growth. But, and this is the big but, the traditional ways of finding these antibodies, they've always had some major drawbacks. For starters, they take a ton of resources, and they're slow. Really slow. You're often looking at things like big animal immunization campaigns or these massive screening experiments where you sift through maybe thousands, sometimes millions of potential designs.
Millions. Just hoping to find a few good ones.
Exactly. Just hoping. And even when you find those initial hits or leads, they usually need months, sometimes more, of downstream work. You have to optimize their affinity, basically how tightly they stick to the target, and their developability. That's, making sure they can actually be turned into a stable drug that works in humans.
Okay, so it's not just finding a binder. It's finding the right binder that can actually become a medicine.
Precisely. And maybe the most frustrating part, traditional methods often just fail completely with what we call hard targets. These are really important spots on proteins, maybe crucial for a disease, but they're just tricky to hit, structurally awkward or hard to reach. Traditional discovery just struggles there.
So when you paint that picture, the huge effort, the cost, the sheer amount of trial and error, the hitting walls with hard targets, it really feels like the field was crying out for something different, right? Was there this sense that, okay, we need a better way, not just want one, but need one?
Oh, absolutely. You could really feel that. We've had computational approaches trying to help for a while now, but honestly, their hit rates in the lab actually finding something that worked rarely got above maybe 0.1%.
0.1%.
Wow. Think about that. Less than one design in 1,000 actually pans out experimentally. So even with computers helping, you still had to rely on these massive high-throughput screening experiments in the lab. It was still a huge search.
Kind of defeats the purpose of using computation if you still have to do all that screening, doesn't it?
Exactly. It undermined the whole point. You were still basically looking for a needle in a haystack. Maybe the haystack was slightly smaller, but not much.
Okay, and that sets the stage perfectly for Chai-2. This is where it comes in, promising something fundamentally different. And the core idea, the buzzword, is this zero-shot capability. What does that actually mean? Because zero-shot design sounds, well, almost impossible.
It does sound a bit like science fiction, right? But zero shot basically means Chai-2 can design potential binders for any specific spot you point it at on a target protein, entirely without needing a known antibody to start from. It doesn't need a template.
Ah, okay. So unlike older methods that maybe just tweaked an existing antibody.
Right. Chai-2 generates completely new functional antibodies from scratch for a target or an epitope on a target where it has never, ever seen an antibody bind before in its training data. The paper describes it as an innovative multimodal generative model. Multimodal means it can handle different kinds of data together, like protein structures, sequences, binding information, and generative is key. It doesn't just analyze, it creates new things. It represents a big step forward in what they call all-atom generative modeling.
I like that.
Meaning it understands and models the interactions right down to the individual atoms. This lets it predict how the antibody and the target will fit together, the whole complex, with really high accuracy.
Okay, and this is where it gets really interesting because the results they report are just staggering. They state Chai-2 achieves a 16% hit rate. 16 in fully de novo designing from scratch. For people who follow this space, what does that jump from, you said, 0.1% up to 16% actually mean in the real world?
It's massive. It's an improvement of over 100-fold, more than 100 times better.
Over 100 times.
Yeah. Just try to picture the time, the money, the resources that went into finding just one working antibody the old way. Now, with Chai-2, well, if the old methods found maybe one good hit out of 1,000 tries, this model is finding like 160 in that same batch size.
That's incredible. It changes the economics completely.
It really does. It's not just a slightly better number. It feels like a fundamental shift in how you can even think about tackling drug discovery projects.
So it's not just faster discovery, potentially, but maybe making it viable for targets that were just considered too difficult or maybe too niche before.
Exactly. And the speed aspect is also key. The paper says with this high success rate, they can do the experimental validation, actually testing the designs in the lab and characterizing the good ones in under 2 weeks.
2 weeks. Down from months or years.
Yeah, it's a drastic compression of the timeline.
Yeah.
And it links back to the efficiency.
Right. They mentioned the workflow usually involves asking Chai-2 for maybe, up to 20 designs per target?
That's right. Maybe 20 antibodies or nanobodies, which are smaller antibody fragments. And often, that handful of designs fits into a single standard 24-well lab plate.
Okay, so what does that mean practically, fitting it onto one small plate?
It means you can often just skip the huge, expensive, robotic, high-throughput screening step altogether. That's usually a massive bottleneck.
Ah, I see.
A 24-well plate is tiny, fits in your hand. Being able to test a small number of highly promising candidates like that, it makes the whole process faster, cheaper, and honestly more accessible. It could really open things up for, say, rare disease research or other areas where the cost of traditional screening was just prohibitive.
That really is a game changer for the workflow.
Yep.
But, okay, impressive numbers, impressive speed, but did it actually work in the lab? What about the real-world experimental proof? How solid is that?
Yeah, good question. And the paper really goes into detail on the wet lab validation, which is crucial. So they tested Chai-2 against 52 different targets. These were diverse targets and, importantly, novel targets.
Novel meaning?
Meaning there was no known antibody or nanobody structure already solved and deposited in the big public database, the Protein Data Bank, for any of these targets. So these were genuinely new challenges for the AI, and the result, Chai-2 successfully designed at least one working binder, one hit, for 50% of those targets. Half of them.
Half is pretty good for genuinely new challenges.
It's very good. And it showed versatility, too. They specifically mention a 68% success rate in designing something called mini proteins, which are these small, stable protein binders. And these weren't just weak binders. They routinely got picomolar binders.
Picomolar. That sounds strong.
It's incredibly strong, like molecular superglue, often hundreds or thousands of times tighter binding than what you might initially find using older methods. That saves a lot of that optimization time we talked about earlier. And maybe the most striking example they gave, Chai-2 managed to find the very first computationally designed antibody hit against TNF-alpha.
TNF-alpha. Isn't that a really major drug target?
Yeah.
For things like arthritis?
Exactly. Hugely important target for inflammatory diseases. But it's notoriously difficult for computational design methods because its binding site is kind of flat and has certain chemical properties that make it tricky. Previous in silico attempts had largely failed. So for Chai-2 to nail this one, that's a really powerful demonstration.
Okay, that's convincing. But one question that often comes up with AI, especially generative AI, is it really being creative? Or is it just sort of, stitching together bits and pieces it saw in its training data? How novel are these designs?
That's a critical point, and they specifically looked into that. The paper confirms that Chai-2 isn't just memorizing or regurgitating things. They analyzed the structures and the sequences of the antibodies Chai-2 designed. And the vast majority are structurally and sequentially novel.
Meaning they look different from anything that's already known.
Exactly. Different from known antibodies in the databases. They are genuinely new molecular entities, not just clever copies or mashups. It really is generating new solutions. And another important piece, it's not just about hitting the target. These AI-designed antibodies also seem to have good drug-like properties right out of the box.
Like what kind of properties?
Well, we mentioned the strong affinities, the picomolar binding, but also things like low polyreactivity. That means they don't tend to stick randomly to lots of other things in the body, which can cause side effects. And they also looked at immunogenicity scores, which try to predict if the antibody might provoke an unwanted immune reaction in a patient. The Chai-2 design scored well here too.
So they bind tightly, specifically, and look like they might be relatively safe and well-behaved as potential drugs.
Yes, the profiles look comparable to existing therapeutic antibodies that are already successfully used in patients. That's a huge hurdle cleared early on.
That is fantastic. Because just finding something that binds isn't enough, right? It needs to be potentially usable as a therapy. Okay, so if it's creating novel, usable binders with this kind of success, how controllable is it? Can scientists actually tell Chai-2, okay, I need an antibody that does this specific thing? Can they tailor the output?
Absolutely. That's another key strength highlighted. Chai-2 isn't presented as some rigid black box. It's just described as a controllable, model-driven framework. Researchers can use specific prompts instructions, essentially to guide the design process towards particular goals.
Like what sort of goals?
Well, for instance, designing different formats. Maybe you don't want a full-size antibody. Chai-2 can be prompted to design smaller formats like those mini proteins we mentioned, or VHHs. Those are single-domain antibodies people call nanobodies, or scFvs, another compact format. And really importantly, you can tell it to target a very specific region, a specific epitope on the target protein.
So not just hitting the protein anywhere, but hitting it in a precise spot that you know is important.
Exactly. They gave an example with a target called CCL2. They prompted Chai-2 to design binders for two completely different epitopes on CCL2, and it successfully designed both VHH nanobodies and scFv antibodies for both spots with hit rates of 20% and 25%. Pretty impressive specificity control. And there's more. The model can even be prompted to design for cross-reactivity.
Cross-reactivity, meaning it binds to more than one thing.
Right. Specifically, binding to the same target protein but from different species. This is actually super useful in drug development. For example, they showed it could design a lead antibody that binds strongly with nanomolar affinity to both the human version of a target protein and the cyno version.
Just, like cynomolgus monkeys, the ones often used in preclinical testing.
Exactly. Finding an antibody that works well in both humans and the animal models used for safety testing can save a huge amount of time and effort. You don't have to develop separate tools. So thinking bigger picture, this kind of programmable cross-reactivity is a massive advantage. It could help develop single drugs for diseases caused by slightly different variants of a protein, or just dramatically streamline that whole preclinical testing phase.
Yeah, I could see how that would remove a major headache in the development pipeline. It really feels like we're moving towards much more intentional design.
So thinking about that grand vision in the paper, it sounds like they believe this Chai-2 approach could genuinely reshape the whole strategy for finding biologic drug leads. What's the core shift they're pointing to? The core shift is moving away from discovery based on, well, chance and massive numbers, that sort of stochastic, large-scale experimental screening, towards what they're calling intentional, deterministic molecular engineering.
Deterministic engineering.
Right.
Meaning designing it right precisely from the start.
Pretty much. Designing the molecule you want, atom by atom, to do the job you need it to do.
Yeah.
It's a shift from finding something that works to engineering something to work. And they argue this could unlock drug targets and therapeutic approaches that were just out of reach for traditional methods. It might finally make some of those previously undruggable targets druggable.
Wow. So thinking ahead, what could this enable? It sounds like maybe we could start designing much more complex therapies almost on demand. Could we, say, generate specific binders for precise epitopes to build things like antibody-drug conjugates where you attach a toxin, or bispecific antibodies that bind two targets at once?
That seems to be exactly the potential they envision. Having this ability to rapidly generate highly specific, well-behaved building blocks, these epitope-specific binders could massively accelerate the development of those more advanced, multifunctional biologic drugs. And the paper even hints at potential beyond antibodies and proteins.
Really? Like what?
Well, because Chai-2 reasons about interactions at the fundamental atomic level, the underlying framework, they suggest, could naturally extend to other types of molecules. Things like macrocycles, peptides, maybe designing new enzymes, perhaps even impacting small molecule drug design down the line.
So the ultimate vision is almost designing drug candidates computationally that are so good, so well profiled, they're practically ready for clinical studies almost immediately, like a single shot from computer design to the clinic.
That seems to be the long-term aspiration, yes. Shortening that incredibly long and arduous journey from an idea to a medicine. A future where computation doesn't just assist discovery, but truly drives it through precise engineering.
It really does feel like we're watching a pivotal moment unfold here. This shift from mostly discovering drugs through laborious trial and error to actually engineering them at the atomic level. It makes you wonder, what impact will this have on the future of medicine? And, how quickly might we actually see these kinds of zero-shot design drugs making their way into our hospitals and clinics?
And it also raises a fascinating, maybe slightly unsettling question too, doesn't it? If we're getting this good at designing complex biological molecules like antibodies with such precision and speed, what other parts of biology might become programmable in the near future? And as our power to engineer life at this fundamental molecular level grows so rapidly, what kind of ethical or societal conversations do we need to start having?